Hi all, welcome back. We're going to continue talking about vision. We left off last time we were talking about the structures of the eye. So we're talking about the muscles, the lenses, um, spots on the eye like the macular or the macula. Um, and now we're going to talk about how light refraction works, how the lenses can concentrate the light or diffract the light depending on the structure or the shape of the lens. And then we're going to talk about how cones and rods work and how kind of an image is made off of the wavelengths of light that are coming to the eye. So first, let's talk about refraction. Refraction is the bending of light. And so this happens when light passes through different media. So if light's going through the air and then it goes into water, it will refract or bend. If it goes through the air and it goes through, say, a semi-transparent membrane, and then into a semi viscous liquid, and then through another membrane, like through your cornea, through the lens of the eye, and then into the retina region. Okay, all of this can cause light to bend and change its angle. Okay, and that's really what refraction is about. Okay, the idea, the hope, and kind of the design of the eye, the evolution of the eye, is to take that light that's coming to the eye and concentrate it. Okay, so make it so your lens of your eye is able to focus the photons into a single spot, and that single spot is the fovea centralis. Okay, and that's part of the macular or the maculus. It's the center portion of the maculus. Okay, and so that's the point where the majority of cone receptors are. And so this is the point where if you're trying to focus on a small object or detailed object and it's got details of colors and things like that, that's where you want the most photons hitting, is so you can best see that object. Okay, so convex lenses will concentrate the light waves, so it'll converge the light waves into kind of a single beam. A concave lens will diverge or spread the light beams. Okay, and so there are kind of different purposes for different uses of the lens. Like if you're trying to focus on something very fine, like if you're reading and you're focusing on letters, then you want the lens to be convex and concentrate on a single image. If you're focusing on kind of a landscape and maybe trying to let as many senses come in, as many different colors come in. Maybe it's fall colors and you're looking at the leaves of trees and whatnot. Then your lens is more concave and it lets the light kind of penetrate more receptors than just specific receptors of the fovea. Refraction in the eye, most of it occurs on the cornea or across the cornea, and so the cornea itself is convex. Okay, and then when it does so, it's going to move through the lens, and then it will be concentrated into the retina. Now, interestingly, because of the way light travels and the way it gets refracted off of the lenses of the eye, the image that is produced, at least the initial image that is produced, is actually upside down and reversed, left to right. And so it's very similar to a microscope. If you've ever used a microscope, the image is upside down and reversed in a microscope because of the lenses that are associated with it. Okay? In the case of our brain actually takes that, our visual cortex takes that and corrects that reversal. Okay? So you really never see that unless you have an issue with the visual cortex, and it's not able to do that. All right, so if we look at kind of what we're talking about is a convex surface, so whether this is glass or a lens or whatever it might be, will take the light that's coming in. These are just photons that are traveling on wave patterns, okay? And it'll take it and it'll converge it into kind of a single region, okay? If it's convex. If it's concave, okay, then it causes the light to actually diverge or spread out. Okay, and so our eyes, for the most part, are convex, so the light is going to come in and kind of form a single image in many cases. Okay, and so that is going to form, in this case, it's showing you the image is actually upside down because when the light comes through the cornea, it reflects, and it reflects in the opposite direction. So the base of this tree is reflected up here, and the crown of the tree is reflected down here. So the image is actually upside down. And then when that signal is sent to the brain flips it back over. All right, refraction disorders. When this happens, okay, there are situations where individuals and maybe you're one of these individuals, you have a disorder, disability, a malfunctioning of certain parts of this refraction capability. Maybe it has to do with the lens, maybe it's the cornea, maybe it's the retina, maybe you have macular degenesis, so it's the maculae area. There's all kinds of issues that individuals can have with their eye. We're going to talk about a couple. Okay, so presbyopia or farsightedness. Okay, typically this is because the elasticity of the lens, so the flexibility of the lens, the ability of the lens to flex and come back and flex and come back, it decreases over time. Okay, and so when you get older, you lose that elasticity. Okay, and because you lose that elasticity, you have the inability for that lens to flex back and forth like this to change from being able to see far away to seeing up close, etc. You lose that power. And so often reading glasses are used so you could focus and at fine kind of details, and that will provide that refracting power that's needed. Nearsightedness or myopia, okay, is when the eyeball is actually a little bit too long. Okay, and so the light focuses, it comes in and it creates kind of a blurry image. And so one way to do this is use a concave lens, a corrective concave lens. And so that could be a concave lens of like eyeglasses. It could be the concave lens of a pair of contacts, or you could actually have it corrected with kind of something like LASIK surgery, where you're actually correcting the myopia. Okay, and then farsightedness from a hyperopia. Okay, that means the eyeball is actually too short. So these are typically genetically controlled. There might be, well, there's some genetics involved here, but this is just due to the elasticity overuse. Okay, and so some individuals, they never experience this. Okay, so they have good genes, their elasticity stays, but others individuals, it's just, you wore it out, okay, and now you need some reading glasses. Hyperopia, on the other hand, though, this is due to an actual eyeball that is too short, okay, and so now you have blurry images for close objects and you can see fine for further objects. So instead you need a convex lens, okay, so that you can see things up close. And astigmatism, okay, is a defect in the curvature of the cornea or the lens. Okay? And so it's a defect in a region. Some region of the cornea or the lens has a defect. So it's not, the curvature is not perfect. And so you can correct that with things like glasses. So one part of the glasses, the lens of the glass will be a little bit, depending on the stigmatism, it'll be a little bit more convex or more concave, okay, and that would correct for that region of the eye that happens to be have astigmatism. So let's look at this a little bit. So if your eye is too long, okay, that means that when the light comes in, the light overshoots kind of the point of focus. The point of focus would happen prior to the fovea, the central part. Okay, and so it would overshoot, and that refraction would carry out further, and that will cause a blurry image. Okay, if your eye is, where's it at, too short, okay, then the point of focus would be way back here. So again, you're getting wave patterns that are not concentrated enough at that focal point. Okay? And there are ways at which you can correct that. Again, we talked about this. Concave lens, okay, would cause that focal point, okay, to be corrected if it's too far or if your eye is too long. A convex lens would correct for an eye that's too short. Okay. All right, let's talk a little bit about the photoreceptors. Okay, photoreceptors come in really two main types. You have rods, okay, and they get their name because of their shape. Okay, and they also have a pigment in them called rhodopsin. Okay, and so to remember rod, rhodopsin, that's pretty easy. Okay, they're associated with each other. That's a light sensitive pigment, but not a light sensitive pigment from a color perspective. Okay. It's really what allows us to see or to distinguish shapes, and shades of gray and things like that, especially in low light, dim light. Okay? So it's really what you use when you're trying to distinguish things in the dark. Okay, you might see the outline of something. That's really your rods firing, and it's not just the dark. I mean, it can happen in many cases. It's how you see gray and different colors, black, whatnot. The cones, on the other hand, which in many cases get the most time spent on them and most concentration, is because they really are what allows for us to see color. And that's what allows for us to distinguish not just shape, but texture, size, I mean, distinction of different regions of an object, these kind of things. So it really provides the sharpness or the ability to distinguish fine details. That's where the cones come in. And we are trichromatic. Trichromatic really means that we have three different light sensitive pigments, okay? Erythrolabe, chlorolabe, and cyanolabe, okay? Now, other organisms might be something else. You might say, well, dogs can't see a certain color, okay? So maybe they're dichromatic, okay? So they only actually have two of the pigments. And then if you might say, well, things like mantis shrimp, which can see far more colors than we can see. They can see infrared, they can see UV, they can see all kinds of things. They're actually pentachromatic. They actually have 10 different sensitive pigments. So for the most part, we're trichromatic, although there are some individuals, mainly females, in the population that are tetrachromatic. So they actually have a fourth pigment. Okay, and yeah. Anyways, we can talk about these kind of things, or you might have an assignment on some of these things. So this is what they kind of look like. Again, there's not too much a difference between rods and cones, kind of morphologically, except for rods are typically longer and cones kind of have a more cone shape to them. So here you can see the rods, here you can kind of see the cone shape to them. But in general, they look very similar to each other. They function very similar, except for they react to different photon lengths. So different wavelengths of light stimulate the different cones and the rods. Okay, and so that photon would be coming. What's not shown here is light's coming in from this direction. Okay, it's going to react to the rods. The rods are then going to create a stimuli that creates kind of an action potential, and that action potential will then be transmitted through the nerve fiber to the optic region of the brain, regardless of whether it's a rod or a cone. Same kind of general system. Okay, that sensitive pigment. Okay, if we dive into that rod cell and look at that rhodopsin, okay, that's that region that is very sensitive to light. So it's sensitive to photons that are coming in. Okay, when it reacts, okay, rhodopsin itself is going to decompose into opsin and retinol. That's where the name rhodopsin comes from. Okay, is those two molecules combined, okay, form or opsin and retinol form rhodopsin. Okay, when penetrated by light, okay, these molecules break apart. They decompose. Okay? In doing so, that can trigger a reaction. Okay? And that reaction, along with the energy that's associated with the use of mitochondria, can cause the synaptic ending to release a impulse that is going to then initiate the nerve to fire an action potential, or to have a action potential. Okay, adopsin is the light sensitive pigment in cones. Okay, so a little different, and that really depends on what type of light pigment we're talking about. Remember, I said we are trichromatic, so we have three: ithrolab, chlorolab, cyanolab. Okay, and this is why we often say, well, we have primary colors, and we can distinguish certain things. And actually, when we look at the visual light range that we can see, so if we look at what we call visual light, it runs from about 400 nanometers. That's blue light wavelengths. That's where cyanolabs are going to pick up. Okay, and it runs all the way to about 700 nanometers. Okay, that's where red light picks up, and at about 550 nanometers is where green light is picked up. Okay, so across the visual spectrum, okay, the visual light spectrum, it's based on our eyes and our capability of picking up different wavelengths of light. And so it's really based on these three molecules and how they pick up different wavelengths. All right, let's talk about some other unique kind of capabilities of our vision. So stereoscopic vision, okay, which really provides us this great capability of, because we are bifocal organisms, so we have two eyes facing the same direction in the front, okay, we actually have stereoscopic vision, meaning that our eyes cross and see a single image. And that single image, that allows for us to judge distance. Okay, and the nice thing with that is, probably through evolution, instead of having eyes on the side of our head like a lot of other organisms do, which then they have a little bit different type of vision. They have more of a peripheral vision, so they can see things to the side, and they don't have very good vision necessarily in the front of them. Okay, they're not bifocal, but they have the ability to sense maybe predators coming from the side, things like that. Our eyes are really geared for the ability to focus on a single object and know how far that object is. And that really probably had to do with maybe hunter-gatherer capabilities of primates. Okay, so the fact that the right eye and the left eye can focus on a single object, send that refraction back into the eye, and then our brain generates a single object, single view, okay, allows for us to judge lots of things. Okay, mainly distance is probably what it evolved at, but it also allows us to judge the shape, the height of the object, the width of the object. Is it a 3D object? Okay, is there a backside of the object? These other things that maybe eyes that are not bifocal or stereoscopic can do. Yeah, so really just more like overlapping images that really allows for you to determine the three-dimensional image of the object. So you know that there's also a top and a bottom to the object and a backside to the object, because your eyes can see both regions or all regions. Okay, so the visual pathway kind of works like this. We're not going to dive too deep into this, but you'd have photons that are coming in through the cornea, through the lens, okay, picked up by the cones and the rods. Those nerve fibers are going to cross. So your right eye crosses over to the left hemisphere. Your left eye crosses over to the right hemisphere. Okay, and so that normally is called the retina crossing over. Okay, and that really allows for the optical lobes to generate an image, a uniform kind of image, a single image. Yeah, I don't know that I want to talk too much more about that. It'd just be diving down into the weeds. You guys don't really need to know the other parts of this, but just knowing that, the fibers cross, that this crossing, at least a portion of the fibers crossing, allows for the stereoscopic image to be made. So it allows for basically the left side and the right side to kind of be merged into a single image, and the importance of that. It also happens to be involved with that flipping, that flipping of the image back over and back from a left to right deviation or crossing. Okay, and yeah, I mean, in general, it just helps with a lot of things. Depth perception, so how far is something away, and this is not the same kind of tracking that we see in organisms that have their eyes kind of more on the side of their head, like maybe a cow or a horse or something like that. You don't see those optical nerves cross like this. They're forming separate images from each side of their body. Okay. All right. Let's talk a little bit about kind of the last kind of lifespan changes that have to do with sensories in general. Of course, hearing loss. Hearing loss is big. Most of us have elderly parents or grandparents, and that you know are related to hearing loss. There are a couple ways at which hearing loss can happen. You can have damage to the hair cells from over stimulation. So if that individual worked maybe in a factory or something like that, and continuously stimulated those hair cells with very loud noises, you can get tinnitus, which is going to be that overstimulation of hair cells. Along with that, you can get degeneration of neural pathways to the brain. Okay, we got age-related visual problems. We already talked about the hearing a little bit, but we get age-related visual problems. Of course, you can get other things that affect the lens, so you can get dry eyes that affect the clarity of the lens. You can get floaters, which are going to be crystallized, typically things that are related to like calcium carbonate, little teeny crystals that form in the humera or in that liquid-like substance, and they kind of just float around in there. And so if individuals see spots that float or they have floaters, that's where that comes in. The elasticity of the lens is a big one. Okay, so especially with age, that lens, its flexibility it's just gone, or its ability to retract and come back to its original shape, it's gone over time. Glaucoma, cataracts, macular degeneration, all of these affect different regions. Okay, the macular degeneration, the maculae, is that kind of yellow spot in the eye where you have a high concentration of cones, and it's really a major focal point of the eye. And when you have degeneration, you have breaking down of that material. It's not able to focus as well, and so you typically lose the ability for fine details. And there's just other things like that have to do with the lens or the cornea of the eye. And then age-related things that have to do with like smell and taste, that other sensory. Okay, you can just have olfactory receptors that are lost over time. Okay? They're overstimulated, or they've been damaged from different things. They could. We kind of talked about this a long time ago. Your cells have a certain limit on how many times they can replicate. Okay, that's called the Hayflick limit. And so you only have a certain amount of times that you can replenish your cells. And in many cases, if it's a neuron or a nerve cell that's damaged, although the olfactory, we already talked about it, they do have the ability to reestablish themselves, but only a certain number of times. And so if it's a neuron and it gets damaged, normally it's not replaced. And so over time, if you've done things to damage olfactory or visual neurons or neurons that are associated with the ear, taste neurons, etc. If you've damaged them, often they won't be replenished, and you can have damage to that sensory, okay? Or just overstimulate them. If you're constantly working in an environment of smell and taste, okay, you can overstimulate them, and then you get less of a response, especially as you age. Okay. All right. Next time out, we're going to talk about the endocrine system and kind of how glands work and how hormones work.